to you today. So Emmanuel is coming to us directly from molecular and cellular developmental and neurobiology at the University of Albany, as you can see on his screen. He is a PhD candidate and is here primarily because one of his buddies, and you'll hear a little bit more about that, said, hey, we should have him come down and talk about zebrafish since we haven't had any zebrafish talks. And so we're very excited about that. Emmanuel got his master's and bachelor's degrees from the University of Ghana. And for his master's degree, he worked on kombucha and gut microbiome. And so that's another topic that we would all be very interested in, and won some really nice awards for that work. And so we're excited to have him here to talk about zebrafish, but hopefully there's a little kombucha in there too. Or we'll talk about it.
Before we get started, I'm trying to remember one of the things I'm supposed to do at the beginning. We just want to give you this back and appreciation for you taking the time to come down and speak to us.
So thank you all for coming in for the seminar talk today. It's one of my, I think I said to Carly, it's my first professional speaker as a scientist. So I gave a similar talk at ASM, but this will be a bigger story because it's a longer time. I'm in my fifth year of my PhD with Alex Brown. Yeah, today I'll talk to you about how we are working to dissect gut microbiome, spatial structure and function, particularly in our zebrafish model. And we will know that hundreds of microbes live in us as humans and animals. And these can influence various aspects of development, right? The factors that shape the composition of these microbes and how they are established along the gut is not properly or hasn't been fully mapped, even though we have ideas of what microbial diversity might exist within those structures. But really having this tool to be able to visualize the native microbiome in this ecological ecosystem, the native state, allows us to be able to start teasing apart essential microbe interactions that drive specific host responses and be able to visualize how these would give us insight into understanding host-microbe interaction, host-microbe dynamics. So just a little bit about myself and my journey, and just a recap of how I got to meet Samuel is that Samuel and I were in the University of Ghana from 2012 to 2016, and that was when we graduated from our bachelor's degree. I did biochemistry and he did animal biology. So it was there that I started research, and my first research was studying Babesia infection in cattle and seeing how infection changes over time and how that impacts cattle health. Then with that interest, I went to do a 1-year postbac training, and that is why I started the microbiology journey. So here, my goal was to characterize the microbial community that ferments kombucha. And so I did nanopore sequencing and did metagenomics, and I characterized a plethora of genes, microbes that were present in the SCOBY, that fermented kombucha. So that was exciting for me, and I wanted to take that a notch higher. So now I freeze-dried the kombucha and then tested that on anti-diabetic mice to really see if the kombucha played a role in ameliorating diabetes and also plays a role in shaping the gut microbiome. So this is the thing that I studied in for my master's. So with my microbiome experience, studying how the kombucha changes the gut microbiome, I joined Alex's lab in 2020, right before the pandemic happened. So in Alex's lab, we actively study microbiome spatial organization, how microbes interact in their complex environment and using imaging techniques. So most of the work that I'll show you today is deeply rooted in imaging and developing new imaging algorithms that can help us to really just dissect this microbiome informations. So just what I want to accomplish today is to show you how we think about scales in microbiome studies. We think about information along a scale, from a bigger scale to a micron scale, and show you two evidence of from my research work where we see that variability in the gut microbiome really alters host phenotype, and show you an improved machine learning enabled spectral microbiome that I developed in my PhD work for dissecting or identifying microbes in the community of constitutional microbes. and show you what that I did to start looking at specific microbe interactions in a binary system to see how a pathogen interacts with probiotic organisms. And then I'll end with highlights of other works that is done in our lab, especially in the oral microbiome and canine oral microbiome and spatial imaging technologies. So I started off with discussing microbiome across multiple scales. And with the HMP projects, which most of you will be aware about, that studied different microbial ecosystems across different sections of the human body. And I want you to take note of the scale bar that I put here. There's a meter scale bar. So we're looking at organs or regions that are distant from each other of the human body. And you will see that these are really well-clustered microbial communities that exist across different sites on the human body. So when you look at the scale of the human body, you see the spatial assembly or spatial organization of microbes across different sites of the human body. You could see that within each animal, within each individual, there's inter-human variability that existed in the sampling of these healthy individuals. But overall, we see the spatial clustering of prepared microorganisms that establish unique ecosystems across different scales. So take note of that scale over here. And in another study where they looked at different squirrels, and here they looked at the axis of the GI. So they looked at from the small intestine to stomach to small intestine, large intestine, cecum, and then the fecal samples. And what you could appreciate is that within each column in this heat maps here, there's different abundances of microbes within those sections of the gut. But the take home here is that along the centimeter scale of the longitudinal axis of the gut, there is a spatial distribution. And this is remarkable and might have profound impact on how these microbes function in their host. So when we want to study microbiome, we have to think about the holistic approach to studying microbiome. And we think that our model systems allows us to really study this native assembly of microbes in the GI because it's an accessible system and it's we can image the full length gut axis. So this scale here describes what that there's another level of variability that can exist along the axis of the gut. So in 2021, we wrote this review article where we described the biogeography and bioarchitecture. And we described the biogeography as a macro scale structure that comprises of microbial patterning that is driven by host ecological factors. So when you look here in this graph here, in this image here, you see that along the small intestines through to the rectum, there is this heavy increase in microbial density shown here. And this microbial density is characterized by changes in host physiological conditions that selects for different bacteria to establish across different sections of the gut. And even within the same sections, in the villi of the intestines and in the lumen, preferentially, different host factors drive this assembly of microbes and distribution of these factors. In the oral microbiome space, where this image is coming from, because the oral microbiome is really accessible, people could take dental swabs and they could image with dental swabs and start seeing at the micron scale specific microbe interaction that exists in these oral cavities. And in this image from Jessica's lab, they start really unraveling specific microbe interactions shown here in different colors, that each microbe has this relationship that it exhibits or it is used that allows it to interact effectively with another microbe to form these really conserved structures that I was studying in the human microbiome. And these are profound for understanding how the oral microbiome is established and how changes in the oral microbiome can lead to disease and illness. So we have the tool that can allow us to really start mapping specific microbe interaction that exists at the scale of the micron scale within these microbial world that exists within us. So what I wanted to highlight in this slide is that the mechanism by which intestinal microbes interact is oftentimes poorly mapped and just characterized as an unstructured continuum of chemical resources. So we think that microbes will just move in tropism to chemical resources, but I think that there's more to that. We think that specific microbe interactions shape this assembly in addition to host responses. When we talk about eubiosis to the state of dysbiosis, oftentimes we describe that as a loss in community abundance. We say, okay, there's a loss of community from a healthy to a disease state. What we really don't talk about is that is this shift in community associated with changes in structure within the communities. And what I'm showing you here in this image is that when you see these two images, you could see that there's a loss in some particular members, leading to loss of some structures that existed in the community. But some structures are preserved. And the question that we want to ask is that, do those preserved structures serve the same function in a dysbiotic state? It's something to think about, because if it's a structure that is required to function, then if it's lost and the structure is still preserved, we should have the same function. So we start thinking about dysbiosis not only in loss of compositional abundance, but also in structural partnering and structural organization. So we think that several factors shape material composition as well as shape shape microbial communities. So with the advances in optics and microscopy, this has allowed us to be able to magnify specific microbial interactions and then really start understanding their role in the context of their ecosystem. So why do we care about spatial structure as a lab? And we really think that the spatial organization of microbes is important and is required to establish and sustain metabolic agents and biophysical interactions that define unique host microbe interactions. And by understanding this underlying structure that exists in the microbiome, we can start thinking about questions like, what is the mechanisms by which these microbes interact? What are the rules governing their assembly? And how can we really manipulate and disrupt this structure and study different aspects of host microbe interaction, and also study interspecies dynamics and aggregation. And I just showed a figure of work by Alex, my advisor, and in the oral microbiome, they have these networks that they can really start teasing out symbiotic relations that exist across multiple microbes that exist in the oral cavity, and how they form the dental plaque to perform their functions. So this, as I mentioned earlier, leads us to really start thinking about model system that we have in hand and how we can really utilize our model system to the best of our advantage to study these microbial interactions and structures and organization. And one of the advantages of this zebrafish larvae are optically transparent, so they allow us to, it's amenable to imaging, so we could really see what is in there in their native ecological state. And then we can generate or manipulate this conventional zebrafish that has a natural microbiome to generate lotobiotic models that we would assemble unique consortium of microbes and then start probing with a sub population of microbes, start probing functional roles of these microbes in the host. So we can generate this tractable lab, controlled lab experimental models that allows us to really start probing mechanistic studies of microbiome assembly. And these also have high breeding rates, so we can generate this colonies with ease and fast. Just a limitation to lotobiotic models is that when we generate those models, we, in essence, simplify the natural microbiome, right? And the question is that does that oversimplification alter the function? Remember, I said that we want to see if the structures are conserved. If they are conserved, might they have a role in performing the same function, even if they are reduced complexity? So we have to think about the fact that, or know that we have oversimplified the microbiome and think about whether that will alter the function that we expect. And there can be variability in how these colonize the gut based on how you introduce the microbes to the gut. You could expect variability. And how do you really test your efficiency of recommercialization to see whether you successfully reconventionalize these models and generated the models that you idealize in your experimental setup. So the birth of my work started with this preliminary observation where I want you to take really critical observation of this data in that GF is germ-free and FC is a pathogen, which is F. pulmonari, it's a Frischmann pathogen. And when you treat a germ-free mice with a pathogen, they rapidly succumb to the infection and they start dying. In the conventional zebrafish, there's protection with or without the pathogen. I mean, with the pathogen's protection. If you generate a mixed 10, not a bad model, that has 10 bacteria as well from the conventional zebrafish, you are able to restore protection against the infection. But if you generate monoclonalized versions of these, you lose protection, except for chrysio. But if you pull back this 9 together, you're able to restore protection. So we start seeing that fundamentally there's community level protection that's offered by the microbiome and individual protection. But as a lab, we're interested in how this big community of complex microbes interact to offer this protection. So our hypothesis was that the spatial structure that exists in our 10 and 9-member community allows for these biochemical and biophysical interactions that leads to differential host responses, especially to the pathogen, but in general, host responses. So one of the very early experiments I did was I was trying to see, can I visualize these microbes in the gut? And if I can I observe some structures that existed in these communities? So I did this 3D rendering of confocal image where I label for bacteria with universal bacterial probe and perform fluorescence in situ hybridization. And what we started observing is that bacteria can exist within the GI as larger aggregates, single cells, smaller aggregates, and have this spatial distribution along the GI from the bulb region through to the distal region. And we started thinking, what microbes could be in this larger aggregate? What microbes could live in this smaller aggregate? What microbes could live as single cells within the GI? So that was our initial observation, and we wanted to really drive home with this and start thinking about how we can design in situ probes that would bind to each of these targets and be able to identify them. So, what we did again was then to see, okay, now that we've seen the spatial partnering that exists in the microbiome, can we study this across our different conditions and see if we see a kind of structure that existed there? In the conventional zebrafish, we start seeing that there is this high abundance of microbes that start off in the bulb region of the gut and then move as the bacteria get expelled through to the distal regions of the gut. But fundamentally, we see that there is this conserved, heavily colonized bacteria that existed in the bulb region of the gut. In our mixed 9 model, we see 3 phenotypes where there's heavily colonized microbes, there's medium, and there's the least, suggesting that what is driving these spatial changes. Yeah. Which microbes are there? And note, this is just a single level for all microbes. And we expect that they should have 9-member communities, but we see different structures. Does that suggest that the different microbes that move in and out of the gut at different times? So we started thinking about so many questions that existed in our mixed 9 model, really thinking about how we have some preserved structures, but we lose some structures, and how these is all playing out in our experimental model. And it was good for us to note the source of variability that in our mixed 9 model, that its origins from microbiota resides in the gut. So maybe there's some host factors in the gut that allows for system microbes to establish right there. So we start thinking about Host-Microbiome as one of the physiological markers in the host that can allow for bacterial spatial partnering in the gut. So we thought, how would the existing resident commensal microbes affect host ability to secrete mucus? And how will this mucus also affect the resident microbes ability to establish and attach and bind and have these egg and chicken storage. Which one does what first? Is that something to think about? But we know that the mucus provides nutrients for residential microbes and provides this scaffold material for biophysical attachment, and also allows for signal molecular cues that can drive bacterial ecosystem or bacterial establishment in the gut. So we wanted to understand the role of microbes in modulating mucus and the role of mucus in modulating microbes establishment. So again, in our experiments, we observed that in the conventional zebrafish that has a natural occurring microbiome, we see this really nicely arranged intestinal mucus lining that lines the epithelial walls of the gut with high densities. But in our germ-free models, we see this mucus patches that are now properly assembled in the gut. The gut loses epithelial membrane integrity, and we start seeing that even along the proximal gut, there's this few patches of mucus that is not really well established. So we think that the microbiome plays a role in triggering or sensitizing the host to secrete this intestinal mucus, one for the host protection and also for the bacterial gain for adhesion. So we're excited about this observation and wanted to just probe further on this. So again, we looked at this across mixed 9 germ-free and conventional models again, and we see that here is mucus in grayscale and bacteria in grayscale. There is highest bacteria in conventional with highest mucus, and least bacteria with intermediate mucus, and then the least in azenic, as you would expect. So when we quantified these images and we perform analysis on them, we observed that when we plot a graph of bacterial biovolume to intestinal mucus intensity on the y-axis, we see this real well separated cluster of mixed 9 bacterial to mucus, where there's high bacteria, there's essentially high mucus in the conventional fish. But in our mixed 9 model, we see that there's variability. Where there's low mucus, where there's low bacteria, there's high mucus. Where there's high mucus, there could be low bacteria. And that is captured in this graph here. So we didn't see a correlation that existed in our mixed 9 models with high bacteria corresponding to high mucus, because we see cases where there's high bacteria and there's low mucus, where there's low bacteria, there's high mucus. So we start thinking that maybe specific microbes are required to drive these host responses. And in that sense, we thought that was something we could go down that road path to strand and level. And again, I said this already, we quantified the mucus, and it was highest in the conventional, intermediate in the mixed 9, and the least in the azenic model. And I said that the bulb in the azenic was disrupted. So that's something to take note of, that the gut loses its integrity when there's no microbes. So we wanted to really start understanding if specific microbes were required to sensitize hosts or required for host mucus secretion and played a role in host mucus host microbe interactions, let me say it that way. So we know that for the mucin, which is the peptides that are in mucus, these are all glycosylated, and this sugar moieties, or this peptides with the sugar moieties interacts with resident microbes with receptors that allows them to anchor themselves to the mucus layer on the host and start assembling their communities. So we know that there's a receptor-mediated interactions with bacteria and mucus. And in Karen's lab, they disrupted some of the mucus receptors on the bacteria, and they observed that once you do that, they lose their ability to adhere to mucus. So we start thinking about that, the thought that for our model, we propose that in our conventional zebrafish, where there's high bacteria and there's high mucus, we think that there's something occurring that's natural, and that we think that the host and the microbe have co-evolved to have these relationships going on. But in our mix-9 model, what we observe is that there are regions where there's higher bacteria and low mucus, and where there's low bacteria and high mucus, and where there's high bacteria and high mucus. So we see this variability over there. So leading us to hypothesize based on our model that mucus stimulation in the gut may be more dependent on combinations, more dependent upon the presence and distribution of specific microbes rather than total biomass. So we think that it's not only about how much is there, but it's about who is there, and it's about who is there with who that is driving this particular host responses. So that's something that we wanted to complete this session with. So I'll move on to talk about our approaches that we have developed in our lab to be able to start really mapping out which microbes exist in these communities and are found at each of these various specified locations. So with sequencing, you can resolve and discriminate closely related taxa by sequencing, even to the strain level. When you sequence, you can really find difference in taxa, but you generate low spatial information because of the approach. Whereas with imaging, it's hard to discriminate these close spaces because you design 16S fish probes, and these target regions on the DNA that is really close, and so you have no room for mismatches to make effective probes. But if you do that, you're able to generate a high resolution spatial information about microbial organization. And just to give you an idea of the microbes we have in our mix-9 consortium or mix-10 consortium is that we have bacteria that are closely related in strain and species. So we have a lot of Pseudomonas and a lot of Amonas in our setup. So we have to design a really effective imaging approach to be able to discriminate these organisms. So our imaging approach starts off by building a database with our 623S genes, RNA sequences, and then we design lists of predicted probes. We validate their efficiency to be able to bind to their single target and check how efficient they will hybridize, all in silico. And then we validate these probes in the lab, in vitro, and then try and use them for our analysis. So in an ideal situation, what we would expect is that each probe will bind to its target and not to any of the off targets. And then we'll generate a matrix of all our 10 probes and then we can assign them with different fluorophores, and then we can image. Simple. Experimentally, that wasn't what we observed. We observed that even when we try to design this highly specific probes, the probes bind to their off targets. And as a PhD student, this was one of the hardest moments for me, because I was like, oh, this is a 2-week experiment. Do this and we'll be good. And I do this after several months, and then it's looking like that. And so we were stuck on that result for it took us 2 years to figure out a way around this. But we were stuck on this experiment. So after several back and forth, should we repeat? Should we design new probes? What should we do going forward? We observed that within each of the cells, that is within each column, there is a unique combinations of off-target and on-target binding that were in each of these cells, right? So if we can regenerate this ID, it's a fingerprint of cross-reacting probes and target probes within each cell, that can become an ID that we can use to identify each of these cells within the matrix and say, this can be classified as cell A and cell B. So that was what we thought about. And we're like, okay, we'll see what we'll do with this. So I presented there's 2 problems, which I'm simplifying here. When you perform imaging where you have multiple fluorophores, and I'm talking about fluorophores between 10 or 6 and 9, you are going to select fluorophores with emission spectra that fall within the range of your detection limit of your microscope, right? So we have a wavelength constraint. So you have an inherent problem of fluorophore crosstalk. And if the peaks are not well separated, it becomes inherently hard to identify or to unmix those fluorophore signals as true signals for one cell. So this is inherent. We have to deal with, we know we have to deal with this. And then we have it convolved with a new problem of probe non-specificity. And the goal over the last 2 years was to be able to design an approach that would simultaneously solve these two problems at the same time. So we thought about machine learning, and we thought that if we could really identify those signals and we produce those signals, those can become input for training data sets, and those classifiers would be used to those patterns and identify those patterns as this fluorophore with this combined fluorophore signal. So we took that as our home run that would use machine learning and then use our ideas on the fluorophores that are there and our knowledge on the cross-reactivity pattern that we know that would exist there and the differences that exist in those fingerprints for our training data sets. So what we did is that we made this cocktail of all the probes with our different fluorophores across our spectra. And we make this cocktail and then we treat them. So each cell will get a cocktail of all the probes. And our hope is that when they get into the cell, there's going to be a differential combinations happening within each cell in those tubes. And hopefully those are unique enough to enable us to discriminate those. So we did that experiment, we obtained our images, and then we have to now think about processing and think about how we're going to do that. And I'm a biochemist, not a computer scientist. So it was a learning curve right there again. So we had to think about dealing with noise in the image, right? And being able to minimize the noise to improve our training inputs, because garbage in, garbage out. So we thought about reprocessing the image in such a way that we would reduce the noise but not compromise on spatial resolution. Because if you can't tell that this is 5 cells apart as one cell, then you're losing information, you're losing data. So we wanted to still be able to reduce the noise in our image, but also be able to preserve the spatial information that existed within each image, because we really wanted to find and start finding specific micro-micro interactions. So at every at the little scale, we are interested in those changes and being able to tell them apart. So what we did is that based off of that combined spectra of fluorophores binding within a cell, on the spectral dimension, we can generate a spectral peak that is not for a single probe, but it's a combined spectral peak for all the combined probes within the cell. So we expect that for each of these cells, we will generate this unique heat map that is a unique ID of combined spectral information within each of the cells. So if you look across each column, each row here is the unique spectral combined spectral information that existed within each cell, shown as a heat map. So each of these rows seems to be very different from each other for our training set. So with that, we trained our model, and our model came back to us with 99.8% accuracy in detecting true pixels from non-specific pixels. So we are just about 0.2% off in accuracy. So with that in mind, we're able to transform this matrix back into our idealized matrix. So that was right before I went for ASM. So that was really a good one. So we did this, and then we were excited about this output and wanted to find out if we build a synthetic community, we have this classifier, can we go into a community and then identify each of these microbes in there? So I did the experiment to put all the cells together, put my cocktail of probes in there, and then applied my classifier. And we were able to identify each of the cells that we put in there in our artificial community. So this is the individual cells, but this is a mixed community, and we're able to identify and classify all the cells within our community with such high level accuracy. and with about 84% sensitivity, looking at how much you put in and how much we recovered across multiple fields of view. So this was really, really, I think this is one of the highlights of my training till this point. But we were able to do that, and we wanted to see, can we again do this in vivo? Can we take our classifier and go in vivo and see if we can identify microbes within the zebrafish gut? So we did that, and we applied our classifier to our zebrafish gut, and we observed that we are able to identify microbes within the zebrafish gut. And here we identified there's 3 microbes. But what was really good for us was that in pink was Eremonas, right? Magenta is Eremonas. And from the sequencing, Eremonas is also the most abundant microbes in these fishes. So we start thinking about our analysis and our work in the context of, is one microbe sufficient to drive a response in the host? And in these communities where we have the conventional and this Notabag models, is it predominated by just one microbe? What other microbes? How can we really account for the other microbes that are present there? What are they doing? And we start thinking about several ways to manipulate our system to reduce the levels of Eremonas and see if we can still pump in more microbes. So we're in the process of doing some of those experiments where we can alter Eremonas concentration and see if we can build the diversity further. But we start thinking about the community, thinking that maybe one microbe is what is, or several microbes is working in the different regions. But that was just that we can apply our classifier in vivo, and it's worked really fine. So the future for this work is really to start teasing out specific microbe interactions that exist within the gut, because we have the tool in hand, and really start looking out for host factors and look out for active goblet cells and see if regions where there's high mucus, there is really high activity of goblet cells, or it's a distantly triggered signal that is sensed. So we want to study different aspects of host microbe interactions over the next few months. So to summarize this other part of my talk, is that we developed a machine learning-based spatial manipulation algorithm that has about 99% specificity and about 84% sensitivity. So that allows us to deal with this crosstalk problem and probe non-specificity problem simultaneously. And that our model is compatible with in vivo applications, right? So we can apply this with several other in vivo studies. But the lessons we've learned going forward is that the image processing step is really a critical step in determining the best trained model. How you segment the image and how you pre-process the image is critical. And you need to generate this large data set. So we have this 82 channel image, which really gives us a lot of variability that we can record in terms of for signals. And you don't need to design probes that are specific. You don't need to kill yourself to redesign the experiment. You can see if you can reproduce patterns, and then you can think about machine learning as a tool to really harness the differences to come up with a tailored approach for your particular applications. So I'll end with our work on Chrysio, where I showed you that Chrysio is the only monoclonalized version that is able to offer protection. So we wanted to think about how this is doing that individually, how is it doing it. But I'll just show aspects of imaging data just for your curiosity, but not any other part of the data. So the first question I wanted to ask is, does Chrysio or the CMAS, does it interact with this pathogen? Is there any form of interaction that existed between the pathogen and then the probiotic protective bacteria in this case? And what we observe is that when we look at the zebrafish, the whole fish mouth, we see that this Chrysio CMAS is on the gills around the same place where the pathogen will bind in a control experiment. So this is just CMAS, which is just a pathogen on the gem-free fish. They preferentially bind to the same site around the gills, which is where the infection usually is established. But when you have a fish that is pre-colonized with CMAS, and you have flavor coming in, you see that for each of the flavor cells, there is this a ring of or ambush of sea moss driving to where the flavor is and trying to contain the flavor.
So cool.
But what you see is that when you have a fish that is colonized with sea moss, the amount of flavor that could bind the fish is significantly reduced. Which is shown in this graph, that if you have a sea moss fish, the number of pathogens that could establish on that fish is significantly reduced. So we think that the mass is actually colocalizing with the flavor, but also reduce the number of flavor that can infect or attach to the host, thereby reducing potentially a mechanism of reducing mediating infection. And then we looked at the skin mucus, right? We started thinking about what is the skin mucus on this fish. And we saw that in our gem-free, the skin mucus is higher in our monocolonized with sea moss than our gem-free. So we think that sea moss is also probably triggering the host skin mucus, maybe for adhesion, but we think that's a phenotype that is being driven, that is absent in the azenic zebrafish or the gem-free zebrafish. So other aspects of this work involves talking about or finding out how the zebrafish responds to both of them, and if we can generate mutants that don't have the ability to protect, and see how those are also interacting with the fish. So to conclude my talk, for the host microbe interaction study, we think that there is a dynamic relationship between the microbiome and the host mucus. That is not a function of presence or absence. We think that there's more to that. We think that there's a spatial relationship among specific taxa that drive a spatially graded host response, in this case mucus, and drives differential gene expression along the GI of the gut. And for our tech advancements, we think we have demonstrated that we have this machine learning based spatial medicine algorithm that allows us to map bioarchitecture in the microbe of closely related taxa. That's the take home, that we can do this even with high resolution in combinations of communities that have really close related taxa, and that is estimated. And CMAS reduces the pathogen colonization and also colocalizes with the pathogen. So that's what I've shared about my work today. And just the last 2 minutes, I'll talk about what other people are doing in our lab. And just Teresa works on studying oral microbiome, and she's interested in studying how binary interaction between Cyanobacterium and Aedes aegypti exists within the microbiome. And she starts seeing these Aedes aegypti preferentially binding to one end of C. microcodite, and they start forming this premature dental plaque. And when she treats this Cyanobacterium with heat-killed Aedes aegypti, we start seeing that there's a shrinkage in the morphology of these Cyanobacterium. So we think that there's these contacts or binary interactions that really mediate or leads to different microbe interactions within the gut. And we see different expressions of different genes such as fluotinin and antitoxin in coagulations with Aedes aegypti than Cyanobacterium alone. And something that some of you are interested in is we work also on canine microbiomes, and Nicole, who is leading this project, studies biofilms in canine and how that relates to periodontal disease. We think that's a good model system for studying periodontal diseases. And she starts seeing really fundamentally beautiful structures that are similar to what we saw in human oral microbiome. We think that there's shared spatial information that might exist within these communities. And Alex Linus, who just published this protocol paper where he designed this in vitro dental plug on the chip model, where he has this flow cell models where he cultures dental plug, and he images them and sequence some of them, and starts seeing that the donor plug has this remarkable heterogeneity in the taxonomical of the microbes. And when he seeded these microbes and he looked at the plant from the donors, he sees small and large aggregates of microbes. And he thinks that those are the things that nucleate these larger diverse communities. So he has setups where he disrupts the communities in the dental plaque. And when it disrupts them, he loses the diversity of microbes. So he thinks that those aggregates are important to nucleating this big architecture of dental plaque, not single cells, just as the species, single cell species biofilm models predict, but I think that this little baby fragments are the pioneers that established these communities. And Rogue was a mathematician in our lab, and he also really designed this sparse low rank Poisson correlation and regression and mixing model for unmixing, and this is the least square approach, and this is his approach, and you can see that his approach is probably superior to identifying microbes in the dental plaque compared to the existing least square approaches. So we are working all around the clock, especially in imaging, to really start using our microbe interactions in our host. With that, I'll thank members of my lab and Alex, my PI, our collaborators in France, Pastor Rebecca and David, and Noma and Pastor, who are postdocs. And Alex wants me to invite you guys that he's hiring a postdoc, and I'm also looking for a postdoc, so it's vice versa. He's throwing me out here, and he's asking if any of you want to join our lab. You can come in and join our lab in Albany. Thank you.
Looks like we have a few minutes for questions, and as usual, the first question we'd like to come from a trainee.
That was a great talk. Thank you so much. I am so curious to know if there are fungi or yeasts in zebrafish guts.
I don't know,
Okay.
to be honest. We haven't tested that, but I should think there should be, but I don't know.
Thanks.
I could find out.
Zebrafish is such an excellent model in the lab, but is much known about the microbe ecology outside of the lab.
Again, I don't know,
Yeah,
because I haven't studied environmental interactions with zebrafish, but these are just lab experiments.
Yeah.
But I would, so the point is zebrafish live in this aquatic medium, right? So they don't live as humans in the air interface. So they are in the water, right? So these hosts environment is different from a human host environment, right? So we think that that issue can be happening in the environment. And this microbe just move around the clock, just get in and get out. And these are done in time, so just recycled in and out.
follow-up question, if I may. The machine learning model approach is fascinating. And when we do machine learning models on microbiomes to map microbiomes to traits, it's multi-dimensional, it's many features going into that mapping. And one of the things you can do is extract out feature importance from those features, determine what's most important in the analysis. Is that something that can be applied to the kind of microscopy machine learning models you're building to inform us about what are the features that matter most in terms of building accurate imaging? And that seems like a lesson we can all learn from. Maybe at some point, the group could put something out about what is most valuable when we're thinking about microscopy and microbial communities.
Right, right. And, so what we are using right now is information that we have about the flow force that we are using. And then the combined intensity. Remember when I gave this talk, someone asked me, can I add shape of the cells to it? That can be an important feature to add, but not in the case where all the cells look the same.
Yeah.
So there are other things that we can think about adding, but we start from those that have main control over, because we choose the flow force that we can use. And we try and choose that based off of what the cell should look like. So when the cell is long enough, we could put that two together, right?
Yeah.
Because those are like similar part can sparsely distributed. So we think about stuff like that.
And you can do the same kind of verbal analysis statistically with a machine learning model. So you can actually have it output, what are the most accurate features that are predicting these patterns, right? And that, backed with statistics, could inform the community about what we all should be thinking about when we're doing our microscopy. Thanks.
So I'm really interested in how, when you put in your synthetic community, the variability and how that colonizes, that's super interesting to me. And so I was wondering, have you tested to see if the mucus that is produced in exenic or in these notobiotic fish, is that chemically different? And maybe the microbes cannot utilize it as well as a carbon source as the ones produced with full.
That's a good question. That's something that I think Karen has a paper on some using glycans that she thinks that play what I was in specific microbe binding to each. And she has this paper where she shows that mucus in the lungs or mucus on the skin is different from mucus in the gut. Right, so microbes that bind to mucus in the gut might not necessarily bind to mucus in the skin, and there's this selection for what kind of glycans they interact with. And if you see here, we think that different glycans will mediate different interactions with mucus. So that is possible, that there are fundamentally different mucus that is triggered in these different setups.
A big problem is the production of methane in ruminant animals in the rumen. And I've always wondered about the spatial biogeography of bacteria, archaea, and protists in the rumen, and how they might influence production of methane. And that's something we really have to address if we want to lower, methane emissions. So what, do you have some ideas, or do you know of anybody who's trying to apply these kinds of technologies to biogeography of the rumen? And do you have some ideas about how sampling needs to be taking place to facilitate this kind of analysis?
I know, but it's a good question, right? But I don't know how to advise for sampling, because one big part is fixation, how you fix a sample before you apply this fluorescent dyes. So what I had to troubleshoot a lot about on fixing this zebrafish in a way that the gut content is preserved, the mucus is not degraded. We think about method of fixation as one of the really critical steps. And when you have a rumen, you're definitely going to dissect the gut. Think about how it's going to fall apart. You don't want it to fall apart. But I don't know how that's going to work experimentally, but we can think about it. But definitely Alex might be interested in that.
Thank you.
Question, if I may. So I find this sort of spectral deconvolution to be fascinating. Do you have a sense of what sort of plexity you could obtain? Right? Like, do you know how many organisms you could resolve in theory here? Like, if you could optimally design the set of probes, like, it strikes me that you could probably resolve many, many more than 9.
Yes, so we are aiming for 100. I suppose that Rogo, Alex was starting to be 100. But yeah, that's the goal, but it's kind of challenging because even with the 9, you have to reproduce these signals. It must be reproducible. That's the big part is the signals must be reproducible. So you could start and then scaling it up a little bit. But what I didn't show you here is that in this community, we have, we have just 9 probes. Okay, we have, so we have 9 probes. So we use the same probe for every. So we have the same probe. We didn't design 2 probes. We have 1 probe for this, and then 1 for each other. So we have 9 probes, and we're able to tell 10. So I think that we can design less probes, but really take advantage of the signals that the off targets will build up in the cell. So that we don't have a cocktail of dealing with probes rehydrate among themselves and everything and all those things. And how many fluorophores can you use? That's another thing to think about. You only have a range that you can deal with along the wavelengths, right? So the fluorophores, there's a cap to how many fluorophores you can use. I think you can use about 16 fluorophores. So you have to rely on the combined spectral within each of these. So there's a limit to the number of fluorophores you can use, but not how number of cells you can detect. So you think about it, technically, is the fluorophores that's the limiting factor.
Another.
Have you seen specific structures of microbes repeat within an organism? And if so, would this suggest modular functions of microbes?
Did I see specific structures?
Yes, specific structures repeat within an organism.
Yes. So I saw structures, but Alex thinks it's premature. So I didn't add it to this talk. But I see structures that are conserving, mixed mind, and conventional. But once I can confirm that, I'll go to paper.
Thank you for a fascinating talk. I really enjoyed it. I had two disparate questions. One is, have you thought about studying other kinds of like hospital relevant biofilms with this technique and try to start resolving how are they interacting is one question. And the other question that I had was with that proving that causality between the two microbes, like the pathogen and like the competitive exclusion is just a beautiful story. Have you looked at all at any host factor on that story? Have you looked at all at like what's going on with the host immune system when you have or don't have those bacteria agglomerating around the pathogen?
Yes. So this paper will be out soon. The manuscript's almost ready, but I didn't talk about that because it's part of the bigger project. But actually, we see that there's a lot of toxin inhibition here. So the probiont is able to neutralize toxins of the pathogen. Most directly, it's just make sure that it becomes renders it impotent, right? But we have other RNA-seq data on the host changes. I don't know the full details of that. I did read the manuscript on that aspect, but that's for stuff that we have those data, and I can look that up. We can talk.
Maybe time for one more if anyone has a last question. Right, why don't we thank Emmanuel one more time?